Hi everyone, my name is Nolabiba Natasha binti Mamidzi and I am a final year student in the Bachelor of Science Hans Biodiversity Management program. Today I'll be sharing my final year project titled Skills Characterization of Selected Fish Species of Jengka River Tributaries, Felda Semarak, Jengka 15, Pahang. So, what's the issue here? Okay, do you know the Jengka River is part of the Pahang River Basin, one of Malaysia's largest and most important freshwater systems? But even though it plays a huge ecological role, there's still very limited research on the fish species in this area. Human activities like pollution and land use changes are affecting freshwater ecosystems, not just in general, but also in terms of fish biodiversity and water quality. This is very concerning, and yet we don't really know what's happening in Jengka. So that's where this study comes in. So for this project, I had three main objectives. First, to look at the scale types and shapes, basically the morphology of the fish species that are found in the Jengka River tributaries. And the second one, to analyze their scale measurements, or we call it as morphometrics, like length, width, and radius. And lastly, to figure out the most unique characteristics that can help differentiate one species from another just by looking at their scales. Interesting, isn't it? So, okay, here's what I did for this project. Fish were collected from two coordinates of the river. Okay, the species that were managed to catch were Rasbora elegans, Rasbora myrosi, and Rasbora sumatrana. These three are commonly known as ikan seluang, a small freshwater fish people often see in rivers and streams. Then we had Pristolepis gruti, which is usually called as ikan sepat, and Trichogaster trichopteris, which many people know as ikan patung or tree-spot gourami. Okay, then they were caught using cast nets, and once I had the fish, I took their scales, cleaned and preserved them properly. Then I observed the scales under a compound microscope to study their structure, and then I used ImageJ software to measure things like scale length, width, and radii. Then I calculated ratios like L1 over L, L2 over L, L1 over L2, and W over L, and analyzed the data using one-way ANOVA and two key HSE tests to compare the differences between the species. So, what did I find? Morphologically, Rasbora elegans, Rasbora myrosi, and Rasbora sumatrana all had cycloid type of scales. Just to explain a bit, cycloid scales are thin and have smooth rounded edges, while ctenoid scales, on the other hand, have tiny spines or blister-like structures at the outer edge, which is scientifically referred to as a posterior margin. Each species also had one unique trait that stood out. So, what does all this mean? It means we can identify fish species just by analyzing their scales. No need to harm or sacrifice the animal. It also shows that scale structure might be affected by environmental conditions like water quality or temperature. So this method isn't just about species identification. It's also useful for studying ecology and changes the habitat. Okay, why is this important? Because this river, the Jengka River, hasn't been studied in depth before. This project provides baseline data on fish scale types and shapes from that area. And more than that, it proves that fish scales, which most people never think about, actually hold valuable information for science, conservation, and biodiversity monitoring. And personally, I gained a lot from doing this. It made me realize that even tiny things like fish scales can reflect what's going on in the ecosystem. I hope this study helps raise a bit of awareness because, healthy rivers don't just benefit fish, but they benefit all of us too. So, to wrap things up, this project isn't just about studying fish, it's about understanding nature and helping people see the value of the biodiversity. And I really hope it inspires others to pay attention, take care of what we have, and appreciate the ecosystems around us. That's all from me. Thank you so much for watching.